Santham Academy. Welcome to Digital Kaksha. To get updates on new videos, click the subscribe button and the bell icon.
Hello everyone, I am back again with the chapter 5, Principles of Inheritance and Variation. In the last videos we have discussed about the Mendelian genetics, the Mendel's crosses and the Mendel's laws of inheritance. But in the actual world we see that every character is not followed according to the Mendelism. You can see intermediate characters, you can see a tall plant, you can see a dwarf plant, and even you can see an intermediate plant. So it means that in nature the entire characters, they are not transmitted in the way the Mendel have told. Means that in between the intermediate characters are also present, which we can easily see in our nature. So let's see what are the deviations from Mendelian genetics. So in this video we will be discussing about the deviations from Mendel genetics or deviations from Mendelism. So let's start with the first deviation, that is incomplete dominance. As the name indicates, incomplete dominance. That means the dominant allele is not capable of completely suppressing the recessive allele. The F1 hybrid will be showing some characters from the dominant allele and some characters from the recessive allele. So F1 will be an intermediate between the two characters which we have taken or the two alleles of that particular character which we have taken. So let us see what is this incomplete dominance. Incomplete dominance means the dominant is not completely suppressing the recessive allele. So here the F1 is intermediate between the expression of alleles in the homozygous state. Now here the ratio will vary, and second thing is that the F1 hybrid will not resemble to any of the parents. So here the main thing is that the F1 will be an intermediate and it will not resemble to any of the parents, or we can say that it shows the intermediate characteristics between the two parents. So let's take the examples. Here we will be seeing 3 examples. Number 1 is the snapdragon or the dog flower. Its biological name or botanical name is Antirrhinum majus. Second is Mirabilis jalapa, which is commonly called as the 4 o'clock plant. These two plants, they bear red flowers and white flowers. Red flowers, that are the dominant characteristic, and white is the recessive trait. So when the red and white are crossed, we get pink flowers. So it is deviating from the Mendel's laws. So the first example, snapdragon or dog flower, and the second example is Mirabilis jalapa or 4 o'clock plant. The third example in case of animal is Andalusian fowl, which is a type of a bird or it is a hen. Now let us see the deviation from Mendelism in case of Mirabilis jalapa or in case of snapdragon flower. Both of them bears red flowers as the dominant trait and white flowers as the recessive trait. So let us see the cross. Here you can see capital R, so that represents red color flower, which is the dominant trait or the dominant color of flower. Here you can see small r, it is the recessive trait, that means the white colored flowers. So these two will be the parents, this is parent P1 and this is parent P2. Now here you can see that the gametes which are being formed. As both of the plants are in homozygous state, here you can see the gametes which are formed. Here it will be capital R and here it will be small r. Now when the two gametes they combine in the hybrid, we can get capital R and small r. So this capital R and small r, it is the F1 generation or the F1 hybrid. So here this F1 hybrid, it bears pink flowers. Okay, the plants in F1 generation, they bear pink flowers, means neither they bear red color flowers nor they bear white color flowers. So the pink color is an intermediate between the red and the white, or we can say the pink color do not resemble the red and the white. So here the F1 is different from the parents. Now when you self-cross these pink colored flowers, or when you self-cross the plants bearing the pink colored flowers or the F1 generation, here you can see the gametes formed are capital R, small r. Here also the same type of gametes will be formed, capital R, small r. Here this F1, it is in heterozygous condition. Now when the crossing is being done, then you can see that R, it comes to capital R, that means the plant will bear red color flowers. Here you can see the next two combination, capital R, small r, capital R, small r. So these two plants will be bearing pink colored flowers, and here you see the last one, small r, and here you can see that the plant will bear white colored flowers. So you can see that in the F2 generation, whatever offsprings we have got in the F2 generations, they will be the red color flowers, the pink color flowers and the white colored flowers. So here the deviation is from the Mendel's law. You remember that in case of Mendel's ratio, the phenotypic ratio was 3:1 and the genotypic ratio was 1:2:1. So let us see what is the ratio here in case of the incomplete dominance. Here the ratio you can see, 1 homozygous red color flower bearing plant. Here you can see 2 heterozygous pink color bearing plants, and here you can see 1 white colored flower bearing plant. So here the phenotypic ratio you can see that 1:2:1 means 1 red colored, 2 pink colored and 1 white colored. Now coming to the genotypic ratio, here again you can see that 1, here the homozygous condition is there, which is the red colored flower. Again 2, the heterozygous condition is there, which bear the pink flowers, and the third one, that is the homozygous recessive condition is there, which will bear the white flowers. So here the genotypic ratio is again 1:2:1. So here in case of incomplete dominance, the phenotypic ratio as well as the genotypic ratio is 1:2:1. So this varies from the Mendel's laws that in case of F2 generation, the genotypic ratio is 1:2:1 and the phenotypic ratio is 3:1, but here it varies to 1:2:1. So this is how this Mirabilis jalapa and snapdragon flower, they vary from the Mendel's laws or the Mendelian genetics. So here an intermediate, or we can say that the F1 do not resemble to any of the parents. Now let's move to the next example of incomplete dominance. The second example in case of animal, that is Andalusian fowl. It is a type of hen. So here the dominant is the black colored fowl and the recessive is the white colored fowl. You know, when they are crossed with each other in the F1 generation, blue colored fowl is being obtained. Now when you cross the F1 generation, you can again see that you will get the black, blue and white colored fowls in the ratio 1:2:1. So it is similar as we have discussed in case of Mirabilis jalapa plant or the snapdragon flower plant. Here also in case of animals we can see that the F1 hybrid do not resemble to any of the parents, or we can say that F1 is an intermediate between the two parents, that is black and white. So here the F1 hybrid is blue in color as it is not resembling to any of the two parents. Now when we self cross this, you can see we got the phenotypic ratio as 1:2:1, that is 1 black fowl, 2 blue fowl and 1 white fowl. And when we go to the genotypic ratio, it is again the same, that is here one homozygous condition is there, that is the black, 2 heterozygous condition is there, that is blue, and one homozygous recessive condition is there, that is white. So the genotypic ratio is 1:2:1. So in incomplete dominance we have seen that the ratios varies from that of the Mendelian ratios. Here the genotypic and phenotypic ratios both are same, that is 1:2:1. Now let us move to the second deviation from the Mendel genetics. The second type of deviation is codominance. As the name indicates, co means together, dominance means dominant. So together the two alleles, they are able to express themselves when they are present together in an organism. That means that two alleles, they can present themselves independently when they are together present in an individual. So here two alleles express themselves independently when present together. So that is codominance. Now let us take an example of codominance. Here we can see that when we have crossed a cattle with a black coat with a cattle with a white coat, the resultant cattle in the F1 generation has got a brown cattle with black and white patches. Now here you can see that the black and white patches, these were the characters of the parents. So here you can see both black and white color have expressed themselves independently. Here the incomplete dominance is also there. Why? Because the color of the cattle is brown. So it is an intermediate between the black and the white, while the black and the white color has also expressed themselves independently in the form of black and white patches. So this is one of the example of codominance. Now in case of human beings also, the codominance is being seen. The most common example is the ABO blood grouping system. We all know that we have got 4 types of blood groups, A, B, AB and O. Now let us learn something about the blood groups. The blood groups in human beings is coded by the gene I, and this gene I has got 3 alleles, IA, IB and small I. So the blood group, it is coded by the gene I. I means like all the characters, they are being coded by the genes. So here the blood group will be coded by the gene I, and it has got 3 alleles. Normally when you see that according to the Mendel, any gene has got 2 alleles or allelomorphs, but here it is again deviating from the Mendel's laws. Here you can see the I gene has got 3 alleles. This condition or this phenomena in which a single gene has got more than 2 alleles, this is called as multiple allelism. So ABO blood grouping is an example of multiple allelism, and multiple allelism also comes under the example of codominance. So here IA and IB, they are the dominant alleles, means when they are present they will be expressing themselves, and the small I, it is the recessive allele. That means when this small I will be present in homozygous condition, then only it will be able to express itself. Now let us take an example like IA and small I when present together, the A will be expressing itself, or we can say IA will be expressing itself. So the blood group of the person will be A. Second condition, when the two alleles IB and I, small I, are present together, then the IB will be expressing itself and the blood group of the person will be B. Here the two alleles we are seeing, these two alleles, one allele the person is getting from his father and the other allele the person is getting from his mother. So when the two alleles come together, if IA or IB is present, then IA and IB will be expressing themselves and they will be suppressing the small I. So they are the dominant alleles. Now here you can see the third condition where both the alleles, they are the recessive one, small I and small I. So here the blood group will be O. When the two recessive alleles, that means small I, will be coming together in an individual, the blood group of that person will be O. Now coming to the fourth condition. In the fourth condition, the person is getting one allele, that is IA from one parent, and the second allele IB from the another parent. So in this case, as IA and IB both are the dominant alleles, so both will be expressing themselves. So here the blood group of the person will be AB. So this example where both the alleles, both the dominant alleles IA and IB are present together and both will be expressing themselves, so that the blood group of the person will be AB, and this is called as the codominance, means the A and the B alleles both are expressing themselves together independently. So that is called as codominance. Now, the ABO blood grouping can be written as an example of codominance as well as it is an example of multiple allelism. Now let us go to the little detail of about this blood group. Why it is IA, why it is IB and why it is small I, why these three alleles are there. On the RBCs of the individual, a special type of sugar is present. In some of the individual, the type of sugar which is present, that is the A type. That means the allele is IA. IA means it will be coding for the A type of sugar. And if the person has got the IB allele, the IB allele will be coding for the B type of sugar. So when they will be present together, they will be producing both types of sugar, the A type of sugar as well as the B type of sugar. And if A and B, both the alleles are not present, means the RBCs, they are not having any type of sugars on the plasma membrane, in that case the condition is I. That means the small I allele is not able to produce any of the sugar types, neither A nor B. So this is the reason behind why the three alleles are there. That means why IA, IB and small I is there. So this ABO blood grouping system is an example of the codominance, as IA and IB when present together, both will be expressing itself and the blood group of the person will be AB, and it is also an example of multiple allelism in which one gene has got more than two alleles. So in this video we have studied about the deviations from Mendelism. The first deviation was the incomplete dominance and the second deviation was the codominance, where we have studied about the ABO blood grouping system. Thank you. Have a nice day.
Thanks for watching. If you like this video, make sure to like, share and subscribe for more.